Implantation is the attachment and invasion of blastocyst into the endometrium. In this video, I am going to tell you how implantation occurs and what is implantation window.
Hey everyone, welcome back to HM Learnings. I am Harshita, the creator of HM Learnings, where students come to clear their concept and to get the study material. Make sure that you subscribe my channel. So today we are back with the next video on the reproductive physiology, that is the implantation and implantation window. So I'll advise you that if you haven't watched the previous video on the early embryonic development and on the endometrial cycle, I recommend you to watch those videos first before coming and watching this video, because those videos are actually forming the basis of this video. So we will start talking first about the implantation window and then we will switch to the implantation. So here we are actually talking about a cycle in which the ovulation has happened and the fertilization has happened. So we have seen in the video of the early embryonic development that if the fertilization has happened on the 16th day, so from the 16th day till the 22nd day, that is around the duration of 6 days, there are certain changes or certain maturation events which are happening in the conceptus, and these are including the cleavage, resulting in the formation of a 16-cell stage called as a morula. Then there is a process called as a blastulation, which is the formation of a blastocyst, and then there is hatching, which is the zona pellucida is going to be lysed and the blastocyst is going to come out of it, and then the trophoblast can come contact with the endometrium and the implantation could start. So these are the changes which are happening in the conceptus. Now during this time, simultaneously there are changes which are happening in the endometrium. So this is like even if the fertilization happened or not, or no fertilization happened, the changes such as secretory changes and the pre-decidualization, these changes are going to happen. So this is irrespective of whether fertilization has happened or not. So after the ovulation, we know that the secretory phase of the endometrial cycle is going to be started. So this is showing that the endometrium is actually undergoing those changes, which is making endometrium capable of attaching to the conceptus and as well as providing the nourishment to the conceptus. So this is just the preparation of the endometrium to receive the conceptus, and these changes are involving the secretory changes and the pre-deceleration, which is happening under the influence of the hormone called progesterone. So secretory changes such as like they will be increasing the secretions by the uterine glands, which is going to provide the nourishment to the growing conceptus, and the pre-deceleration, as we have seen that the stroma cells, they will form the decidual cells which are loaded with lipids and the glycogen so that they can provide the nourishment when the implantation occurred. And also there is production of the extracellular matrix, which is including the adhesive fibronectin collagen, type 4 collagen, laminin, which is going to make the endometrium adhesive so that the blastocyst could attach. So these are the changes which are happening in the endometrium. So both of these changes are happening simultaneously, they are temporally related, and apart from these things, in the case like this, this is the changes which are happening irrespective of fertilization happened or not till the 22nd day. So in the video of the endometrial cycle, I have already told you that these changes are been happening till the 22nd day, and on the 22nd day, if the fertilization hasn't happened, what will happen? The corpus luteum will start degenerating, and therefore the progesterone level starts declining, and these changes will stop here. But if the fertilization has happened, so the conceptus will enter in the uterus on the 19th day, and the presence of the conceptus in the endometrium on the 19th day is going to facilitate the further changes in the endometrium, such as decidualization and the pinport formation, leading to the implantation. So here, what is decidualization? Like in the fertilized cycle, the conceptus in the uterine cavity is going to facilitate the decidualization, which is the formation of decidua. So decidua is the endometrium of the pregnancy. So there are further changes in the endometrium of the uterus to form a decidua, which is a kind of an endometrium formed in the case of the pregnancy. And apart from this, under the influence of the progesterone, because the corpus luteum is not going to degenerate, so pinport formations are happening, which are the protrusion of the endometrial cells. So protrusion means that if you see that this is the layer of the uterus and you are having the epithelium layer like this. So protrusion means that the stromal tissues are going to push the endometrial cells like this. So now these endometrial cells are they are actually in the more into the lumen of the uterus. So these protrusions, what they does, because uterus is actually being filled with so many luminal fluid because of the secretory activity of the endometrium, which is increased during the luteal phase. So the protrusion is going to endocytose those fluids as well as macromolecules, and this is going to make the conceptus in the endometrium to close to each other. That is the apposition, which is the first step of the implantation. So the thing is that all these changes, the changes in the conceptus and endometrium, they are parallel to each other. They are occurring simultaneously, they are occurring at the same time, and they are facilitating the changes into each other. So these changes are ensuring that the conceptus, the changes in the conceptus is ensuring that the conceptus is able to drive the nutrition from the endometrium, and the changes in the endometrium is ensuring that the conceptus could adhere to it and implant into it. So these changes are actually going to occur only during a specific window of time. These are not going to happen throughout, like there is not a large period of time. Only 2 to 3 days is being there, and this is called as the implantation window. So implantation is basically involving the synchronized changes in the conceptus as well as in the endometrium, and these changes have to be temporally related and occurring in a specific window of time, which is called as the implantation window, which is of the duration of 19th day to 21st day. So this is in the case where we are talking about the normal implantation. In the case of the cycles where where there is assisted reproductive techniques, in that case the implantation window could differ. So it is of the duration, in the normal cycle it is duration of 19th to 21st day. So by 22nd day we are hoping that if the fertilization has happened, the implantation is also going to be completed, and then there is beginning of the pregnancy. So basically after that what happens, because when the implantation process is completed, the invasion is completed, now that syncytial trophoblast, which we are going to see what is that in the coming slides, that is going to start producing the human coronary gonadotropin, which is going to maintain the corpus luteum, because after 22nd day the LH starts falling. So to sustain the corpus luteum, the syncytial trophoblast of the trophoblast start producing the HCG hormone, which is going to maintain the viability of the corpus luteum, and thus the levels of the progesterone. So with this understanding of the implantation window, we will switch to understand what is implantation and what are the steps or stages in it. So there is no proper definition I could get from any standard textbooks of implantation. So it is basically, the definition is basically that it is apposition, adherence and invasion, which are the 3 stages of the implantation. So in the first stage, what happened, that the endometrium and the conceptus, they are lying very close to each other, but this is a very loose contact. In the second stage, like in the first stage there is a loose contact, in the second stage they are going to attach to each other, tightly attached to each other, and in the third state what happens, that now your blastocyst will start entering inside the or invading inside the endometrium. So we are going to see how it is happening. So first let's start from the first state, which is the which is the apposition. So here you see that this is the endometrium epithelium. I turned it into laser pointer. So this is the endometrium epithelium, okay, and this is what is your blastocyst. So apposition means that, that in the video of the early embryonic development, we have discussed that on the 19th day, the blastocyst enters inside the uterus, and it is freely lining in the uterine cavity. So now that this trophoblast or this blastocyst has to come in the contact or come closer to the endometrium, which is helped by the pinpoint formation. So they are going to be come together, and this is the first stage, which is called as a loose contact between the tropovectoderm and the uterine endometrial epithelium. So once they come in the contact, so what is the mechanism by which they are coming in contact with each other? So there are mechanisms which are saying that there are changes which are happening in the layers in the cell membrane of the endometrial epithelium, that the terminal sugars, their structure changes, as well as the zona plexuda has been hedged now. So because of this, there is decrease in the electrostatic repulsion between the two structures, and they could come closer. Another thing is that one of the lining of the uterine epithelium, which is cytocalyx, that is also going to be changed. So these changes are make suring that these two structures could come closer to each other. And another thing is that whole tropoblast or whole blastocyst, all the tropoblast cells, they are having the potential to attach or come in the contact with the endometrium. So they can, from any side, it can come in the contact with the endometrium. So after coming in the contact with the endometrium, what happens at the inner cell mass, there is free rotation of the inner cell mass within the blastocyst. So ultimately, what happens at this free rotation allows the inner cell mass point towards the endometrium at the time of the apposition. So after the apposition, which is just the loose contact between the tropoectoderm, they are closely opposed to each other. They are in a very close proximity. Now, this is just the close contact. In the adhesion, they are going to form an attachment between each other, and that attachment is being done by the help of the ligand receptor interaction. So initially, it was like an ionic bond interaction, that is the electrostatic attraction between the two structures. Now it is going to be attachment, which is involving the covalent bonds with the help of the ligand receptor interaction. So this is going to be a stronger connection between the tropoblast and the endometrium. So via the ligand receptor interaction, and this is involving the receptors of the integrin family. So I'll show here that I try to draw the structure of the integrin so that you can understand more clearly. So suppose this is the tropoblast. So I'm going to draw only few cells. This is blastocyst and this is a tropoblast. And another structure which we are talking about is the uterine epithelium. This is the uterine epithelium which we have drawn. Okay, so this is uterine epithelium. Now I am talking about the structure of integrin. So integrin is basically a bifunctional protein. It is basically an integral membrane protein. So if this is the membrane, integral membrane protein means it is spanning the membrane, it is passing through the membrane. So if this is the cytoplasmic side of the membrane and this is the extracellular side of the membrane, this part is called as the extracellular domain of that receptor or protein, and this is a transmembrane domain of that protein, which is integral membrane protein, and this is the cytoskeleton domain, not cytoskeleton, sorry, cytoplasmic domain or intracellular domain of that protein. Okay, so integrin is also a type of integral membrane protein which is having an extracellular domain, the transmembrane domain, and the cytoplasmic domain. So the cytoplasmic domain of the integrin has receptors for attachment to the cytoskeleton. Okay, and the extracellular domain is having the receptors so that it can attach to the extracellular matrix proteins such as collagen, laminin, fibronectin. Okay, so this is integrin. Now it has two different orientations which could be possible. Like integrin could be present on the on the cell membrane of the endometrium like this, and it could be present on the cell membrane of the trophoblast. Okay, so if it is present on the cell membrane of the trophoblast, what happens? On the inner side, it will attach to the cytoskeleton of the trophoblast, and on the outer side, it is going to attach to the extracellular matrix of the uterine epithelium. This is one possible orientation of the integrin. Another possible orientation is the opposite, that this is going to attach here with the cytoskeleton, okay, and here with the extracellular matrix. These are the two possible orientations. So by this, the uterine epithelium and the trophoblast are going to be contact with each other. So this is the same thing, that it is a bifunctional integral membrane protein with extracellular domain having the receptors for the extracellular matrix proteins, and intracellular domain is having the receptor for the cytoskeleton. They have two possible orientation, integrin on the trophoblast and integrin on the endometrial epithelium. So after this, what happens, that this is ultimately leading to the dislodgement of the uterine epithelial cells from the basal lamina. So we will see here that if we are saying, so I am going to draw again that this is the uterine epithelium. Suppose these are the cells. Okay, these are the cells of the uterine epithelium. Okay, so there is integrin interaction and there is cytoskeleton interaction. This is integrin. So ultimately what happens, that these epithelial cells, they are going to be removed from the basal lamina. Okay, they are going to be dislodged. So there is basal lamina, this is a basal lamina now, which is now accessible to the trophoblast. So here is the trophoblast. So because of these integrin interaction, what happens, that the endothelial cells, they are going to be dislodged from the basal lamina, and now the basal lamina is being accessed by the syncytial trophoblast. So this is the end of the second stage, which is the adhesion. So after the second stage, we move on to the third stage, which is the invasion. So in the invasion, we see that just after the attachment of the trophoblast and the uterine epithelium, what happens, that the cell of the trophoblast starts dividing and they are differentiating to form the two layers. So if I draw here the diagram to show you, so if I'm just drawing the trophoblast, okay, so these are the trophoblast cells, and imagine that they have attached with the uterine epithelium. Now what happens, they start dividing. So they are dividing, that means the number of the trophoblast cells are going to be increasing, and as they are dividing, they are differentiating also. So these layers, the inner layer is going to form the cytotrophoblast, and the outer layer, what happens in the outer layer, that the cytotrophoblast cells, they are going to be fused. So the many cells are going to be fused to form a multinucleated cell layer, which is called as a syncytiotrophoblast. So syncytiotrophoblast is the layer which is actually invading or entering inside the endometrium. So invasion means what? You are actually entering inside that structure. So that invasion is being done by the cytotrophoblast. So the cytotrophoblast does what? It is going to secrete some factors which are going to cause a degradation of the stromal cells. So it is going to secrete the proteases, which is going to degrade the extracellular matrix of the stromal cells, so that it can make the space for the blastocyst to enter inside the endometrium. So here let's see what is being written. So now what happens, it is differentiating into the two layers, the inner layer and the outer layer. The outer layer is a multinucleated fusion of the cytotrophoblast. Then after this, there is the cytotrophoblast is going to invade the basal lamina of the endometrial epithelium and going to reach the uterine stroma. This is being done as the cytotrophoblast is going to release or secrete the tuber necrosis factor, and this tuber necrosis factor does what? This is going to cause a detachment of the endometrial cells from each other. So this is the endometrial cells now. They are going to be detached from each other, and they are going to be detached from the basal laminal also. Then they are going to penetrate and reach the uterine stroma. So after reaching the uterine stroma, it is going to release the metalloproteases and serine proteases to degrade the extracellular matrix and further invade the uterine stroma. So you can see that in this figure, in the previous figure, it was outside, it was in the uterine uterine cavity. Now what has happened in the previous stage of the attachment, you have seen that the basal lamina was exposed. Now this basal lamina will also going to be penetrated by the syncytiotropoblast by releasing the factors, and after that it has reached the stroma. Now after reaching the stroma, it will release the further factors such as the extracellular matrix. To degrade the extracellular matrix, is going to release the proteases, and ultimately it is going to invade inside the endometrium. So now it has entered inside the endometrium. These are the endometrial cells, and now the blastocyst with the syncytiotropoblast is actually inside the endometrium. So now if you know that the stromal cells, they had found the decidual cells. So decidua has been formed. Okay, so if you remember in the first slide, we have said that there is pre-decidualization and decidualization. So they are decidual cells which are present, and these decidual cells are being loaded with the nutrition, the lipids and the glycogen. So they are also going to be degraded, and this is going to provide the nourishment to the embryo. So this has completed the implantation, and after invasion, this syncytiotropoblast is going to release the HCG, which is a LH-like protein, which is going to maintain the viability of the corpus luteum, and the progesterone levels are going to be maintained, and the pregnancy is going to be maintained. So this is about the implantation, which includes the 3 stages, starting from the apposition, adhesion, and then finally the invasion. And the implantation window is just the duration of 2 to 3 days, starting from 19th day to 21st day, and we are hoping that till 22nd day the implantation process is completed. So with this we are at the end of this video. These are the 2 references I have taken to understand this topic. You can look into this for further detail. If you like this video, please like, share and subscribe my channel. Till then, keep learning.